Okay guys, so we are going to do chapter 17 today, which is on viruses. So we're pretty far into the year. I believe after this chapter we only have 5 more chapters to go before we start reviewing for the AP test. This is the shortest chapter of the year, which is kind of nice. So right now in the world, I think viruses are a very big talk, especially with COVID and all the different variations that are starting to come about of COVID. So as you can see down here, here's a bunch of different types of viruses, and these can all infect different types of cells, and they don't necessarily have to be our cells. We could be talking about bacteria and other types of organisms as well. But pretty much viruses, they are just DNA or RNA, some kind of genetic material that is packaged inside a protein coat. Now, they really don't have a cell membrane. The cell membrane for it is just like this protein coat. So it's not like our cell membranes, all right? So a little bit different than our cells. Viruses aren't even considered living just because they are only made up of genetic material along with this protein coat. So here is a picture of a cell being completely polluted with viruses, and all those viruses there are the little red dots. They're just all over the cell. So let's talk about the history of viruses. Back in the 1930s, the first virus ever to be discovered was called the tobacco mosaic virus, and they started working with this, and they did a process called crystallizing this virus. So what they did was they got a lot of the tobacco mosaic virus, and they pretty much clumped it and they froze it. And what this did was it made sure that the virus wasn't going to keep moving around, and we also had a big clump of viruses that were localized to one area so we can study them. All right, and as we already talked about with the blue bullet there, viruses are not cells. They're pretty much just some kind of genetic material inside a protein coat. Now, viruses can be a little different than us. We know our bodies, we have DNA, and that's pretty much all we have that gives us all of our instructions. Now, the DNA for us can go to RNA and turn into a protein, but viruses are a little bit different. They can actually have genetic material that could be DNA, but they cannot have DNA, and they can have RNA instead. And both of these could be double-stranded or single-stranded. For us, it's always going to be double-stranded DNA that leads to all of our instructions. And then our RNA is most of the time, the stuff we actually use is single-stranded. But viruses, they can have all different variations of this. So that protein shell that we talked about, and I just want to talk about the overall structure of a virus for this slide. That protein coat, that is called the capsid, all right? So that's along the inside of this big thing here. That's the pink little, I don't know, dodecagon maybe. But anyway, inside of the capsid, that's where we're going to find the nucleic acids. All right? And how the capsid is made is there are proteins called capsomeres. So the capsomeres make up the capsid. So once again, guys, here is a bunch of different variations of viruses. The tobacco mosaic, our first virus we discovered, that's going to be on the left here. And then all the way to the right, that is a bacteriophage. Now, bacteriophage, just like it sounds, a bacteriophage is a virus that infects a bacteria. So we don't have to worry too much about them unless it infects the bacteria that are in our bodies. All right. So once again, guys, the tobacco mosaic virus, that was our most ancient virus that we actually started to study. Now, there was other viruses. We just didn't study them. But it's made of single-stranded RNA, and it infects many plants. They call it the tobacco mosaic because originally they found it infected tobacco plants, and that's how they found it. All right. But yeah, that one, many different types of plants it can infect. So getting away from plants, the adenovirus. This is something that is more prevalent in humans. So like I said, tobacco mosaic, that's plants. This one it's going to infect babies, young children. It can cause respiratory issues. All right. So, babies might be wheezing, having trouble breathing. Usually it's in the colder months. Sometimes it can actually lead to pink eye. All right. Okay. So here is our bacteriophage, all right, on the right, and I'll show you the whole process with that in a little bit. But on the left one, here's the flu virus, all right, which right now is not prevalent at all just because we've all been wearing masks because of coronavirus. So probably less variants of the flu virus this year. However, the thing that is bad is the flu viruses that are surviving are probably the strongest of them all. So maybe next year flu might be bad again. But anyway, so I will post this video on Canvas for you guys. It's just how viruses invade your body. It's a pretty interesting video, but I'll post that into Canvas for you. Okay, so we're going to talk about what a host cell is. A host cell is going to be a cell that the virus is going to use, pretty much take over, and then use it for whatever it wants to use it for. So it's almost like taking the cell hostage. Now, viruses are going to be let into the host cell. So we are going to have this capsid on our virus, and then around that capsid, we are going to have some proteins, and on the host cell, we're also going to have some proteins. If the proteins on the surface of the virus are going to match the proteins on the surface of the host cell that it's trying to infect, what's going to happen is that host cell is going to let the virus in. All right. If they get let in, that's really bad for us because that means we get infected by the virus. This is exactly how the coronavirus started. So coronavirus, COVID-19, mutated in such a way where the proteins on its capsid ended up fitting perfectly into our human host cells. All right. Now, once they fit in, the virus was able to get into our cells, and then you'll see what it used our cells for in a little bit. But this little mutation that the coronavirus, the COVID-19 virus, that mutation of those proteins on its capsid allowed it to infect humans. If there was never that mutation, guys, we wouldn't have to worry about it. All right. Okay. So bacteriophages. We can also call them phages, and I already talked about this. These are viruses that infect bacteria. Now, there are a lot of bacteria in the world, so there are a lot of bacteriophages in the world as well. So here is a bacteriophage. That's the virus up here. You guys can see there's a protein coat, DNA, maybe RNA, whatever, some type of nucleic acid. And what it's going to do is it's going to take that DNA and push it down through the virus. And this like green looking thing, that is the host cell that it's infecting. So it gets onto the surface of that host cell and it literally injects the DNA into that host cell. Now, when it injects the DNA in there, that DNA actually takes over the DNA of that cell. And it will make that DNA have certain instructions so that that DNA just keeps producing more bacteriophage viruses. And now we got more bacteriophages. And over time, we have so many that the cell is actually going to let those bacteriophages go. And those bacteriophages are going to go into all different types of cells in the rest of the bacteria colony, I guess we can call it. And it's also going to infect them, which causes the viruses to become more and more prevalent. All right. Okay. So as we talked about, guys, the protein on the coat of the virus has to match the protein in the host cell. Given this, every single virus cannot infect just a random host. It's limited to the number just because of that fit that needs to happen. Okay, so once the virus has infected the cell, it's going to use every single thing that that cell has. Pretty much, it took over its DNA, and we know that the DNA is like the main control center of the whole cell. Once it has control of the DNA, it's going to use the enzymes, ribosomes, all the organelles, everything that that cell has, the virus will pretty much take control over. All right. Once the virus has done what it needs to do, the virus will actually exit the host cell, and usually that's going to result in the cell dying. So as the virus is getting into the cell, it's going to fuse its DNA, and we'll talk about this a little bit more in a few minutes, but it's going to fuse its DNA with the cell's DNA. And now we have a whole new DNA in that cell. Now it can choose to do one of two things. It can either make new viruses, or it could pretty much just chill for a while. And we'll talk about why it would want to chill here, like I said, in a minute or two. Now there's two different phases that the viruses can reproduce inside the host cell. So first one is the lytic cycle. Second one is the lysogenic cycle. So I have a video, I'll post a video for you guys on this, but let's start off with the lytic cycle. So this is the one where we are going to have the host cell dying at the end. What's going to happen, and I got a picture for you on the next slide here, but what's going to happen is the cell is going to get infected by the virus. The virus is going to reproduce over and over and over again inside, and then once we get enough of them, the cell pretty much explodes, the viruses go out, and they infect more host cells. All right? So this is called a virulent phage, virulent bacteriophage. All right? So here's the cycle of it in the lytic cycle. We get the bacteriophage, it goes onto our bacteria cell, it infects it, its DNA, which is that little blue thing, fuses with the bacterial DNA, creating brand new DNA to make the baby viruses. Now we got all of these viruses, and after they are all assembled, we pretty much explode our cell and they get released. Okay, so this is pretty much the reason viruses are able to live and reproduce and infect other cells, is through this lytic cycle. Now the other one is going to be a little bit different. You're going to ask yourself, why would we have this cycle? This is the lysogenic cycle. So this is not going to destroy the host cell, right? Most of our phages are going to use both different cycles, okay? So let me move myself out of the way here. When we are using both of these different things, guys, all right, we are going to have, in the end, a bunch of cells getting infected regardless of what's going on. However, if we were just to use the lysogenic cycle, which is the one that does not result in cell death, we're just going to let that bacteriophage just get into the cell, and it's just going to stay there for a while. Now, why would it want to do this? Well, what's going to happen is that bacteria cell, even though it's infected, is going to continue living its life. It's going to reproduce, make more bacteria cells, and each one of those bacteria cells that it makes is going to have that viral DNA attached to it. So here is the lysogenic cycle on the right. Let's just stay with that. So the bacteria DNA is like the darker color here, and the viral bacteria is this blue stuff. As we go through, it's going to fuse with that bacteria DNA, and now we have viral / bacteria DNA, which pretty much the virus DNA takes over everything. It's going to reproduce. Now we got more cells with that DNA in it. And then it can go over to the lytic cycle and start making baby viruses, and then it explodes. So pretty much the virus doesn't have to do too much if it uses the lysogenic cycle first. The bacteria cell's like, oh, I'm fine, I'm good. So the bacteria cell just keeps reproducing and making more cells with that infected DNA in it, And then they all switch over to the lytic cycle, and there you have it. All right. Okay, guys. Last part of this chapter. So we are going to stop here today. I'll continue it. There's only like a few slides. There's like 15 slides left. I'll continue this tomorrow. And like I said, it's a real short chapter. We'll finish it tomorrow, and that'll be pretty much it. So have a good rest of your day, guys, and I'll see you tomorrow.